We're going to be rolling right into how you think about effects for ecological risk assessment at sites. So thinking broadly about the toxicology of PFAS to ecological receptors. Thinking about the wildlife effects, it's generally been observed that there are effects on protein-rich tissues like the liver, the kidney. This is not surprising. PFAS is a bioaccumulative substance and it likes proteins. It's proteinophilic, so it will affect those target organs very readily. Those protein-rich tissues, again, liver, kidney, and cause a cascade of effects leading to usually reproductive, growth, developmental effects that are observable in test studies with the usual laboratory animals. For plants and soil invertebrates, the good news for plants and soil invertebrates is they're relatively insensitive. It takes usually parts per million levels in soil to cause an effect on plants and invertebrates. It takes a lot to cause impacts. The bad news is those animals, those plants and invertebrates will actually absorb and bioaccumulate, as Jen just mentioned in going through the models, and provide a conduit or exposure pathway to the wildlife. And then looking at aquatic toxicity for fish and invertebrates directly exposed via the water column, usually there's a lot of variation in ranges, a lot of high uncertainty right now in some of those values. We'll go through that in some of the information in a few slides. But most concentrations occur really much higher concentrations than your typical human health drinking water concern. We're usually into the micrograms per liter range, not nanograms per liter range for most compounds. So thinking back before the break, Jen showed us the models. Maybe you have a site where you have some tissue data, some measurements in plants or earthworms or fish. You have this exposure information. You can use that to build your usual wildlife dietary exposure models according to the usual ecological risk models that we've been using for the last 30 or 40 years. And at the end of the exposure process, again, you usually have a predicted dietary exposure in your receptor of interest. You have concentrations in soil, water, sediment, surface water. You might be looking at aquatic direct effects, terrestrial direct effects. So really the next question is the exposure and effects evaluation. Do those indicate risks? Are we reaching exposures at the site that might be associated with levels of potential adverse effects? And looking at wildlife, it's the usual evaluation against a toxicity reference value or a TRV, comparing those daily ingested dose estimates that you've modeled using the standard food web models and wildlife exposure models, and comparing those to TRVs associated with an effect or a lack of effect, depending on where you're setting your level of conservatism. For soil life, you're looking at concentrations in the soil, comparing them to some sort of soil benchmark from usually a laboratory study, looking at the effects of PFAS on plants or invertebrates. And then for aquatic life, it's a similar approach where you're comparing measurements in the water to some collection of laboratory studies or a particular laboratory study for an effect and comparing them to effect concentrations. So key question going into an effects evaluation is where to set that level of conservatism. How sensitive should the effects levels be set for evaluating your site-specific exposures? And so for common species, we typically focus on protecting populations of receptors at sites. Usually we're focusing on the low L or no L TRV values. Again, these are concepts that kind of diagrammed on the right, familiar to most ecological risk assessors, but really the effects that are associated with some measurable, demonstrable, very population level effect, like a 30% reduction in growth or 50% reduction in development, 10%, 5%, 1%, where are you setting those levels? That's key to think about. For threatened endangered species, on the other hand, it tends to be more conservative in many cases. Often you're looking at initial benchmarks that may be focused on NOEL levels, NOEL, no effect levels. That's where you're going to laboratory studies and selecting the very lowest dose with an absence of any statistically significant effect, really without regards to the magnitude of the effect in many cases, setting that as an initial kind of layer of conservatism to look at your effects at your site. And you can still consider higher level doses for management purposes. Again, it's a careful and delicate negotiation with your stakeholders, considering your site, the ecology, the animals that you're working with, the exposure pathways, all the uncertainties and things we wrap into an ecological risk assessment. Again, there's no simple answers. For what we put together for the guidance, we focused on really the types of effect benchmarks that are targeting those lower ends. We'll talk a little bit about that in a moment. But the other main question you often have is, which endpoint do you need to look at? Are we looking at development, growth, reproduction, a biomarker of exposure, liver size, delayed predation responses, things along those lines? Which kind of endpoints are we selecting? So in our compilation and review of toxicity information, we tended to focus on the main growth, reproductive, and development endpoints that are used in a lot of risk assessments nationwide. They were the focus of the EcoSSL's review back in 2005, where they went through for many classes of compounds and identified TRVs, again, focusing on those main endpoints. That's not to say other endpoints couldn't be considered at sites. You may have stakeholders or particular organisms or other considerations that may be pushing you towards looking at those other sorts of endpoints. But again, starting with reproductive, development, growth, lethality endpoints tends to be a really common set of practices for ecological risk assessment, and that's where we focused our review for this work. The take-home points, not surprisingly for most ecological risk assessments, you need to think about what TRVs you're using. Don't just pick one randomly. You need to understand it. We put it together a hopefully clear communication process on what these numbers mean, what they represent toxicologically, how we've pulled them from these key studies and put it into the guidance so that you can understand it. Again, as Jen mentioned, all these values, if you disagree, you feel free to dig into the literature, the appendices that we've compiled, the studies, pick your own, reevaluate the literature, kick around the numbers, and really make the decision that's best for your site and best for your risk assessment. Communicating with your stakeholders, again, that key level of effect, which values, how to set that level of conservatism is critical for risk assessments. And at the end of the day, again, we're comparing these exposure estimates to the TRVs and calculating a hazard quotient, an HQ value. It's one number, but take a step back often and try to express that exposure in regards to the benchmarks that you're working with. What's the magnitude of the effect that you're expecting to see? How far are you above that? The hazard quotient, a single number, often doesn't really give you a good feel for what you're protecting, what's the endpoint, and that dialogue is often very useful at some of these sites. So getting to really the focus of the guidance in the effects assessment, really it's all about pulling those toxicity values together in one place, making a best recommendation. We reviewed over 130 tox studies. We considered over 200 values in basically pulling these together. We have recommended values for our 18 target PFOS, focusing on birds, mammals, aquatic life, and soil life. We have 23 TRVs for wildlife, 6 aquatic life criteria, and 4 plant and soil criteria for use in evaluation. I'll go through the simple examples first. So this is the aquatic life risk assessment approach. This is basically a derivation of PFOS aquatic life for freshwater. It was based on a species sensitivity distribution, a similar approach that we basically use in EPA guidance for water quality criteria. It's based on a variety of U.S. resident species, looking at NOEC values, again, very, very conservative, absence of effects, very sensitive. We're focusing on really low levels, very conservative assumptions on threatened and endangered species. And looking at that modeling process, focusing on PFOS, coming up with an HC5 and an HC1 hazard concentration of 1% and 5% based on those levels were in the 0.56 to 6 approximate microgram per liter range per PFOS effects. We also did this for PFOA, and basically that's where we were able to get to in terms of the 18 PFOS. There's simply a lack of information for the other PFOS when you need really a lot of data points to develop this species sensitivity distribution with any confidence. So there are the values for the aquatic criteria we developed. Again, for PFOA, PFOS, and freshwater and marine. The PFOA data for marine are lacking, but it gives you a feel for the levels of where these very sensitive screening levels would be applied. Again, looking at the microgram per liter range for most of these benchmarks. And again, you'd take your surface water concentrations at your sites, compare them to these values as initial screen, and then probably reevaluate further where you need to consider toxicity testing or digging into maybe some of these particular endpoints if you have certain animals at sites you're wishing to protect that may be a threatened and endangered species, for example. Disclaimer at the bottom there, these values are really meant to be a good sort of screening level approach. Again, if you're in exceedance, it doesn't necessarily mean your aquatic life ecosystem is necessarily crashing, that you might want to do some additional considerations, additional evaluation of the data, maybe some additional data collection. So soil invertebrates and terrestrial plants, thinking about the earthworms and the plant effects. Again, good news for the plants and earthworms, they're relatively insensitive. It takes milligram per kilogram levels, as you can see here, to trigger, really get above into some sort of effect level. We had some really good data for PFOA and PFOS where we could develop. There's a lot of data that's lacking on effect values out there, though. Really, so most of what we know are coming from PFOA and PFOS. As far as TRVs, a bit more complicated. There's a lot of literature out there on mammalian toxicity, a lot of information to go through. So we started basically pulling that literature together to focus on NOEL and low-L values. Again, those are the values in studies that are not statistically different than the control response, and the next level above that in terms of dose. Again, the NOEL and the low-L, trying to dial that conservative very low for threatened and endangered species. We reviewed a wide variety of literature. We sent it through a scoring process, again, very similar to what EcoSSLs did for the other compounds that are out there, not PFOS, but back in 2005, looking at a scoring value that was pretty much targeted for PFOS compounds, thinking about data sources, spike versus other sorts of dosing, which analytical grade PFOS they were using, things like that. We scored the studies, we evaluated them, gave extra weight for some of the studies that were focused in on by EPA for some of the mammalian toxicity information used in development of the reference dose for humans, and then also evaluated that really in a best, really what compounds, what endpoints we were focusing in. Again, reproductive, survival, growth, development were the main focuses of our review. And at the end of the day, our TRVs that we've recommended, again, we're not applying any uncertainty factors. So again, if your state or federal regulatory agency or stakeholders want to look at an uncertainty factor application, you'd need to apply those yourself. We didn't have any official recommendations on those because frankly, uncertainty factors are quite a bit variable from document to document. So mammalian TRVs, this is basically the heart and soul of the data product here for TRVs. And what we're showing here, we've plotted these TRVs, PFCAs on the left, PFSAs in terms of low to high fluorocarbon chain length. We got the 3 PFOS precursors on the right, with no values, of course. But for each PFOS, we're plotting the NOEL in the little blue symbol, and then a low-L, the next highest dose if it's available from the particular study we selected. And the little S and G R values either indicate G for growth, S for survival, R for reproductive We tended to pick the sublethal endpoints over the lethality endpoint if it was available. But it gives you an idea of the range of doses for a typical mammal target species where you might see really these benchmarks falling in the 1 to 100 to even 1,000 range orders of magnitude differences. There's a lot of differences in the toxicity of these compounds. There are some indications that once you get to very short chain lengths, for instance, that PFHXA for the PFCAs and shorter, and looking at, say, PFBS on the sulfonates, order of magnitude higher values, which means much less toxic. That's a key factor. The longer compounds tend to have a little bit more toxicity associated with them, which is key, I think, going forward in working with our conceptual site models, knowing which compounds to prioritize in investigation and management. This is the sort of thing that becomes incredibly important. So some of the key challenges that we have is, we're missing data. For example, for PFHPA, the heptacarboxylic acid, we have no values. There's no empirical studies to pull information from. There haven't been any laboratory rodent studies, for example. So what do we do in those cases? You have, as a risk assessor, you have two choices. You either leave it blank or come up with your best guess. Our best guess right now, again, it's a highly uncertain approach, is basically to look at the adjacent acids to see what information is available based on which may be more conservative to look. So we look to the left, we look to the right in terms of chain length, and the PFOA is a little bit more conservative than the PFHXA to the left. So we say a value could consider in modeling PFHPA could be assuming it's as toxic as PFOA. And we have some modeling tools, as Jen will walk through, where you can select that or even put in your own TRV if you feel you want to explore different assumptions in that. It's highly uncertain, but it's a best guess. And that in many cases, that's as best as we can do right now. More studies needed, obviously, but you, as a risk assessor, have to make that hard choice in dealing with the uncertainty and putting these surrogate factors in or leaving them blank and communicating that to your stakeholder. For the three precursors on the very right here, we just went ahead and set those as equitoxic to PFOS because they will, in some cases, transform to PFOS. So that could be the case. So we said, as a conservative approach, you might want to consider setting those at a level of PFOS TRV toxicity. Again, highly uncertain. We need more data. But again, it's better than doing nothing in some cases. And it's a useful uncertainty exercise or sensitivity exercise in your modeling and decision-making process. So bad news for the birds, there's not a lot of data out there. Really limited to PFBS, PFOS. Really, those are the two compounds that have been most studied. We have pretty reliable TRVs we can put forward for those. There is a mixture study where they looked at a mixture experiment with PFOA, PFDA. We've kind of set those in the guidance document as potential numbers that you might consider. Again, a lot of uncertainty associated with those, but again, better than nothing. There are some other ways of looking at dose and effect and linking that relationship. There's been some egg injection studies. So if you have egg measurement data, looking at PFOS in the eggs at your site, that could be a way to evaluate toxicity. We didn't focus in on that for the guides. We were more so doing the traditional dietary dose approach, but looking at eggs could be a way to evaluate that. There is a little bit of data out there on those particular compounds noted at the bottom there. So surrogates, really there's not a lot of good approaches to apply those surrogates, so we didn't really put a good surrogate approach out there for birds right now. So a lot of the compounds where we might be able to model the exposure to birds, we don't necessarily know their toxicity. So what do you do in that case? That's a big question mark. We have some discussion in the guidance. Please look through that, read it. It's not an easy decision point for what to do about, say, if you have some of these other acids present, you need to evaluate the exposures and effects on birds, but what do you do? One potential avenue, again, highly uncertain, is to set it at a very low level, look at PFOS TRV, for example, which is extremely low compared to a lot of the other acids, and assume that these other PFAS are equitoxic to PFAS. That's, again, a very conservative approach in my opinion, highly uncertain, but maybe a potential useful sensitivity analysis to do at your site. Again, we have really all the studies compiled in the appendix, ranked with the scoring, the information about what was looked at in the study. So if you disagree with our selection or recommendation on a TRV, you're free to dig into the literature and evaluate and what you think might be more suited to receptors at your site and more predictive of particular endpoints and other things that you might be looking at. And again, in the model tool, which Jen will go through in a little bit, there's an option where you can enter your own values into the Excel model and explore your own model outcomes. So general observations, the mammalian toxicity data tend to suggest the really short PFCAs and PFSAs are much less toxic than the other ones, the longer compounds for growth, reproduction, and developmental endpoints that we focused on in our guidance review. There's a lot of debate on the potency of the long versus short compounds right now, which endpoints to be looking at, immunosuppression, other developmental endpoints. Really the literature, the science is moving very quickly. I suspect there'll be a lot of really continual focus and reiteration of really what we're looking at here in terms of toxicity over the next few years. For the non-mammalian receptors, that's really a big data gap. We'll talk a lot about data gaps at the very end of the presentation today, but we're missing a lot of the data beyond really the common PFOA and PFOS compounds, for example. And again, good news for the soil invertebrates and plants, they're relatively insensitive, but bad news for the things that eat them. Those screening levels for soil invertebrates and plants generally in the part per million range. And again, the aquatic life values for what we're seeing for PFOA, PFOS, generally in the microgram per liter range. Outside of those compounds, there's some interesting single organism tests for some of the other acids. You could dig into the literature and evaluate those. But again, nothing as comprehensive as PFOA and PFOS to be able to do a species sensitivity distribution like what we put together. But again, all that information is in the appendices and discussed there. In terms of risk characterization, it's the typical, comparing your exposure point concentration or your model dietary dose estimate from your wildlife receptors to some sort of TRV, calculating a hazard quotient. This is standard ecological risk assessment practice. For aquatic life, again, you might compare it to benchmarks, same like you might do for the soil EPCs, the exposure point concentrations you might have. And again, the general framework applies. If you're below 1, usually below a level known to be safe or possibly associated with some level of effects. If you're above an HQ of 1, you're generally above that. Generally I don't stop there. If you're above an HQ of 1, it's best to kind of get back to your TRVs and communicate what you're exceeding. What's the effect you're expecting? What's the magnitude of effect you're expecting if you can get that information from your study? So I'd urge people not to just stop at an HQ step, but really dig into things when you have a HQ over 1, and you really need to communicate what that might mean for your site, your receptors, and really communicating to your stakeholders on what's going on. So we do have that HQ over 1. You might have some unacceptable risks. Then it's oftentimes to go back to the drawing board, not necessarily fire up the yellow steel and go dig up your site, but really think about your risk assessment and consider, refining your exposure point estimates. You may consider some tissue measurements, for example. Again, the model tends to be on the conservative side. We'll tend to overpredict what's in the benthic invertebrates and the fish. Going out and measuring those values might be helpful to your site if it's realistic and possible to do so. The model can help you looking into that model, what it's predicting, which receptors are causing, really resulting in the highest exposures, and what dietary items are driving those exposures. For example, in some work we published in 2018, we did a case study, really 5 case studies of 5 different AFFF sites, applying a model similar to what we have in the guidance, looking at exposures and effects to some key avian species. Again, taking 5 AFFF data sets and throwing them into the model to see what happens. And really kind of allows you to kind of do some interesting evaluation of exposure patterns and doses. But really the end result from the studies with PFOS tended to be public enemy number 1 at these sites. It tended to have the highest exposures of all the PFCAs and PFSAs that we looked at. For some of the receptors, for example, for the spotted sandpiper, you can see some of those bars for those small spotted sandpiper species were in exceedance of the TRV, which means there's potential to exceed known safe levels. So that could be a potential management point for some of these different case studies listed A, B, C, D, E, from left to right there. So we really dug into the model and we evaluated it, and you can see from the model calculations that the main proportion of the spotted sandpiper's exposure was actually coming from the benthic invertebrate ingestion. So an easy thing to do would be maybe to go out to these sites and evaluate what are the concentrations in benthic invertebrates, perhaps do a bioaccumulation study in the lab using some collected sediment to look at accumulation, site-specific BSAF, site-specific accumulation patterns, things like that, to really dial in something a little bit more accurate, maybe a bit more site relevant than the generic model predictions might be. Again, it's something like that you can use to kind of prioritize and evaluate really what are the key uncertainties with your model and where to go next. You know, beyond just looking at additional tissue measurements, you might want to refine your soil, sediment, surface water measurements, maybe get more data collected across the different areas of your sites. If you're limited by data, availability or data quality, maybe it's a good chance to go back and get more data and understand and characterize your site a little bit better. Another potential avenue is putting the eco back in the eco risk assessment, actually going out and doing some ecological studies, evaluating receptors, the populations at your sites, if that's possible, to do some actual observations. Nest box studies, rodent trapping, things like that. There's good ecological studies that you can do to quickly look at the health of the population, before proceeding to active management. Another thing for aquatic life issues or concerns, or soil life issues or concerns, is actually going to collect some abiotic sampling and doing some toxicity testing, seeing if there are actual reproductive or survival impacts in the usual laboratory studies that we use for these sorts of toxicity testing. Basically lines of evidence like that. So really that hazard quotient model step shouldn't necessarily be the end of the road for a lot of these sites. There are other things that you can consider, and often it's an iterative process, collecting more data, returning to your model, of reevaluating it, reevaluating the risk, tightening up the wheels and dials in your model and your site assessment process to arrive at a really sound science and low uncertainty assessment.